Hi, I'm Arran. I'm a research scientist at the Wellcome Sanger Institute, and today I'm going to be showing you about aseptic technique from mammalian cell culture. To do this, we're going to be using a very important piece of equipment called a microbiology safety cabinet, or MSC for short. Sometimes we also call these hoods. We use these to maintain a sterile working environment when we're working with our cells. So now we're going to gown correctly for working in the MSC. After I put my lab coat on, I'm going to put my safety spectacles on. I use a little bit of tape around my wrists to ensure there is no gap in the protection. The first cleaning step for the hood is to use a disinfectant sprayed evenly across your entire working area. And wipe it clean using fresh wipe each time, saturated with 70% ethanol. Now the hood is clean, we need to load the hood with the equipment that we are going to use. Everything must be sprayed with 70% ethanol before it empties. This includes your hands. When we are working inside an MSC, we must always be careful to maintain a sterile working environment. The air in the MSC is being blown down from above, so when we open tubes and bottles inside the hood, we must be careful not to put our hands over any openings and to quickly put lids back on when we are finished working. So now I'm going to show you how to move liquids around safely inside the MSC using pipettes and a pipette boy to help. So if I want to take some media from my bottle, a pre-warmed media, and put it into a tube, I would loosen the lids, load my pipette, removing the lid for the shortest amount of time possible, taking my media, and handling my tubes carefully without putting my hands over the open tubes. We can then use our aliquots of media to fill cell culture flasks using a pipette and pipette boy again. It did not work from stock bottles to reduce the risk of contamination. And that's how you work inside clean MSC.